Welcome, everybody. Thank you so much for joining us once again at this AI 2050 webinar series. You have now entered the final stretch home. We're over the halfway mark, and well done for keeping up to date and keeping with us throughout these 4 days. I know for many of us, it's a massive time commitment. So thank you so much for investing in yourselves and investing in this important work to join us at these sessions. We really, really appreciate it. If you've been enjoying them, let us know. We'd love to have your positive feedback as well. And we're really excited to share this next session with you. And with that said, I'm going to hand straight over to Gemma Turon and Miguel Duran-Fergola from Ursilia Open Source Initiative to take us through this next session. Thank you so much.
Thanks, Nicola. Okay, well, everyone's joining in. Welcome, everyone, and great to have you here in the third session. I'm going to be presenting today, and we are going to be following the same dynamics where you can ask questions in the chat. If you don't get answers right away, we are collecting the answers, and we'll do a longer Q&A tomorrow. So just keep posting them. Okay, so as always, let's start with a few remarks and reminders from the previous sessions to catch up everyone and bring everyone to the same page. I hope by now everyone is familiar with the link, but maybe Jason or Mikel or Nicola, you can drop again the link in the chat where all the course contents are available. And feel free to just keep that open in your tab so you can refer back to it whenever you need, but please stay here with us, look through the screen, and we're going to cover a lot of practical exercises today to consolidate the concepts we've been going over in the last couple of days. So today's goal is to focus on virtual screening cascades and understanding how we can concatenate multiple machine learning models, use more than one model to actually improve our molecular selection, our drug screening efforts, etc., etc. So just as a recap from yesterday, we focused mostly on supervised methods where we have labeled data, meaning bioactivity data, for example, and molecules are either classified as active or inactive, or where we try to build a regression that predicts the specific value of an experimental activity. So this session is going to try and be a bit more interactive, okay? So feel free to drop comments in the chat because we are going to be monitoring them. I'm going to ask a few questions and give you a few seconds to drop answers in the chat, and just to see that you're really active and participative and following along. So all about today is going to understand what we need to be able to act, to use one of these machine learning models, and things that we need to understand, as we saw yesterday, are what are the experimental setups that were used for the training set of this model, how many molecules were screened, that is important, or is this a large screening effort, is it covering a big chemical space, is it small, is it geared towards natural products, towards a particular chemical series, etc., etc. This will allow us to understand whether this model is suitable to our needs. We, of course, need to make sure that it was actually a good model. In the Ursilia Model Hub, again, you will only find models that have already been published and peer-reviewed, or that have been developed internally at Ursilia with tools that have been published and peer-reviewed as well. So you will find mostly good performing models, but it doesn't hurt to double check every time. It's also, for example, interesting to see if this model has been used prospectively by the authors themselves. So they develop the model and then use it on new data, not on a train test validation split only, but actually on new data. How did they validate that the model does work in real life settings, or whether someone else has already used this model in real life settings with good results? That's gonna give you a good head start to know how to use these models. Before we go over, I wanted to quickly talk through some common issues that we need to be aware of. We are not going to dive deeper into those or how to solve them during this course, because of course, again, this will require another entire session. But the idea is that at least you understand these concepts and when you should worry about. So overfitting, as the name says, it's pretty obvious. It's overfitted, meaning it's too adapted to our training set. So if our model is learning from our data at a deeper level, it's learning the noise in our data, it will not be able to generalize, to make good predictions in new data, okay? An example of where this may happen is, for example, when you maybe use too many features that characterize your chemical space so well and so detailed that you characterize things that are actually not important for the activity. They are noise, but the model has learned them, so it will not be able to predict in other kinds of data set, okay? So this is overfitting, when the model is not able to generalize and make good predictions in new data. Pretty straightforward to see. You can evaluate that, for example, using PCAs and UMAP, seeing whether the training with data at least overlaps with your data set or it's very far off. Another issue that we find often is imbalance. In our case, that's almost always when we talk about drug discovery data sets, they are imbalanced towards one class. In our case, mostly imbalanced, meaning we don't have a lot of actives and we have a lot of inactives. We need to be careful about this because even though this is a situation, we need to deal with it appropriately so that our models are equally good at predicting the minority class. Yes, if a data set is too imbalanced, it may only learn about the majority class, it may not perform well. So there is several methods to deal with imbalance. For example, when you oversample the minority class or undersample the majority class, okay? That would be the hints that can get you going into avoiding these issues. The third issue or something we want to learn from the model is its confidence. Not all the models provide this, but it's good to assess whether we can trust the prediction. For example, a prediction on a molecule that falls outside the chemical space of the training data set will have a low confidence of the prediction. The molecule will get a prediction, you will get a number, but you may be less sure about how good this prediction is versus a molecule that is really within the chemical data set. Yes, so confidence allows us to evaluate whether the prediction, we can trust that prediction or not, okay? And then the final concept that I wanted to briefly mention today is interpretability. We talked, some people ask questions about this. Interpretability is very much linked in drug discovery, especially to chemical descriptors that are easy to understand by humans, right? We know why this prediction was made. For example, a mock example, solubility is one of the key determinants of bioactivity. This is one of the reasons why this molecule is predicted to be active. Yes, so interpretability allows us to identify which features are determining whether this molecule is, whether the model is making a prediction of this molecule to be active or inactive, okay? Many models do not include interpretability. Some, as we will see today, do include interpretability measures that help us understand why a prediction was made. Okay, so these are things that we need to keep into account that we didn't have much time to mention in the previous days, so that's why we included this slide here today. So let's go back to our course content. Let's think about this virtual screening cascade concept. The virtual screening cascade is very straightforward. We have a putative drug candidate, and we want to know if it's actually good or bad. So, of course, if we are talking about infectious diseases, we want to know its bioactivity against the target, against the pathogen. We, of course, want to make sure it's not killing human cells. So we want to know its cytotoxicity because more bioactive compounds tend to be more cytotoxic. We may want to assess other ATME properties, other toxicity or advanced properties, like is it going to inhibit cytochrome metabolism, which is going to give me toxicity, or is it going to be metabolized by the cytochrome, so it's going to be cleared fast from my system. Yes, so all these other parameters that make a drug good or not good. If you're familiar with target product profiles, for some disease indications, we do know whether we are looking for molecules that remain for long in the body, for molecules that remain that are cleared fast from the body. Okay, so all these ADME properties allow us to assess whether our molecules will meet these target product profiles. Then we may want to know if we are synthetic chemists and we actually have to then synthesize these drugs to basically test them in the lab. We may want to know whether they are synthetically accessible or not. We may even want to predict the best routes for synthesizing those molecules. There is a whole bunch of models that focus in this direction. And then, of course, we may want to have measures of the chemical space. We may want to do PCAs and UMAPs. We may want to assess whether our molecule is a natural product or not, or is more similar to natural products or not, or is more similar to approved drugs, etc., etc. Okay, so of all these models, yeah, bioactivity is the only one that is truly disease specific. For some of you, we've got some requests, we've got some questions around models that are not for infectious diseases. The Ercilia Model Hub does not have this kind of models. We focus on infectious diseases. All these other models that you do find in Ercilia, of course, are amenable for any disease indication, right? And we are open to including new models, but we cannot dedicate a large portion of Ercilia's time to non-infectious diseases. If there are good models out there that you want to see in the hub, you can come to us and we'll try to include them for you, but we cannot develop these models from scratch, okay? Because that is out of our current scope. Just wanted to make this point in response to some of the questions that we've been getting on the emails. But the Ercilia Model Hub is very useful to do virtual screening cascades, where we can select our compound or our library of compounds, which we use as SMILES. If you check the file that we gave you yesterday, we always represent the molecules as SMILES, and then you can select the machine learning models from the hub that are best adapted to your needs, and basically, for example, in this case, tuberculosis, activity, toxicity, synthetic accessibility, solubility, and whatever it is, to basically help you filter out this library to obtain the final subset of compounds. Again, how do you do this? You can do that through the graphical user interface. The graphical user interface works from the browser through the internet. You need the internet connection. It's very easy, I hope, to use. You just need to log in and paste your molecules in a SMILES format. That's important, a list of SMILES. Otherwise, of course, Ercilia won't be able to process it, but it has a small throughput. As I said yesterday, there is a maximum of 1,000 molecules. Otherwise, the servers will crash. Now we have put a hard stop. So if you try to submit more than 1,000 molecules, the server will not allow you. What happened yesterday is that there was a submission of not the files that we provide for the exercise, but submission of almost 50,000 molecules. So that is well above the capacity of this online deployment that is scaled for small use, for day-to-day use, okay? So I will really ask you, we are going to continue using this graphical user interface for today's exercise. So please use the files that we provide to do the exercise, okay? Otherwise, you will prevent your colleagues from completing the exercise because you will just overload the servers and they will basically not be able to produce predictions, okay? So let's just be conscious about that. On the command line interface side, because we've also got some questions, this works from your terminal, black screen, white letters. So it's for experts. It's people that is not scared of programming, that at the bare minimum, is knowledgeable about Python and Conda. This allows you to do high throughput screenings because, of course, the only limitation is your computing capacity limitation. So you can go up to screening, for example, 1 million molecules, okay? The command line interface looks something like this. You can find a lot more information in the links we provide and on our code and on our documentation, but really it's not the point of today, so just don't jump into it, but feel free to explore it if you know what I am talking about, okay? So today we'll continue to use the graphical user interface to mock this virtual screening cascade exercise so that we get to learn how we could do this and maybe get ideas for our own research. As I said, do's and don'ts for Ercilia at the moment. Ercilia is open and free, and it will remain to be so. You don't have to worry about that. We will always provide our tools for free, so you can keep using them. They won't get closed, they won't get changed. The models that we have there, will keep maintaining them, and we'll keep adding new ones. If you use Ercilia for publications, that's great. We love that. The only thing we really ask you to be conscious is of the original model authors, if it was Ercilia or if it was someone else. So please attribute and cite that correctly. And also, if you used it through the Ercilia platform, attribute it as well. It's important that you just don't get a model and run it without looking through the publication or the metrics about this model, the training data. This is what we are going to be practicing today because we want to make sure you're all equipped to really use this tool for your research. So we are going to be practicing a lot on what we mean by understanding the model background. If you have a model that you want to use and is not on the online deployment platform, not all the models will be there. We'll release the first version in December at the end of the year, but we can add models that you want that are in Ercilia but not for online deployment. We are happy to include them if people want them, find them useful. If you want to do large calculations and you don't know how, you can come to us and make the case and explain your project. And if we have the resources and the capacity, we may be able to help you. This software is provided as is, just a disclaimer. We are not responsible for the models that have been developed by third parties. They have been developed and published and peer-reviewed. So this is what we offer to you. And as I said, we are able to consider incorporation of new models, especially if they are already published somewhere, so the code is already developed, or if there is a curated data set that would make it easier for us to build a model, okay? So just consider whether in your use case there is a model that you want to use that you are not able to, or there is a good data set that you would like to see modeled. In these two cases, we should be able to help you out, okay? So we just want to give you an example of how this is really applied in real life settings. Some of you may have seen this publication from a couple of years ago where we worked with H3D, who had an internal collection of molecules, about 13,000 molecules, wholesale assays for Plasmodium and tuberculosis, cytotoxicity assays for HepG2 and CHO, ADME properties, primary, secondary, like solubility, permeability, and so on. And for a few molecules, even tertiary ADME, what we call more advanced ADME screens, such as cardiotoxicity, okay, which is assays that are more expensive that you typically perform at the end of the drug discovery cascade. We also had data on some enzymology, meaning target-based, not wholesale assay, but focused on specific enzymes. And so what we did is we took all this data and we modeled it so that there is basically a mimic between what happens in the lab and what you can do in the computer. This is really adapted to the H3D chemical space, and they can use now that to prioritize molecules. The paper is quite long, so we don't want to stop into that. But just to highlight one figure is this one here. This is a validation of the model, which is a classification model in this example for tuberculosis to classify them as actives and inactives. It's an imbalanced data set where we have way less actives than inactives, as it typically happens. And this is the result of the classification. You see how the score of inactive molecules tends to be very low, and the score for active molecules tends to be very high, meaning this model is probably good, at least in the H3 chemical space, to separate between active and inactive molecules. Okay, so this is just as an example of how this is really applied to real settings. These models are available in Ercilia. But let's go back to our exercise from yesterday, which is how we can use AI to help us advance drug discovery pipeline against Acinetobacter baumannii. This is a mock exercise. Obviously, we provide you with some compounds, with some curated list, and then what we want to learn here is which models would be useful, how can we read them, how can we interpret the results from these models. Okay. So let's go back to our virtual screening cascade. Before we start with this section, Miguel or Jason, is there any pressing question in the chat that we should be answering?
I don't think so.
Okay.
Yeah,
okay,
thank you.
great.
Good, Scott.
Perfect. So, because we don't have much time in these sessions, we have pre-curated the exercises for you. The models that we are going to be using in this exercise, it doesn't mean you couldn't use others, okay? This is just a selection that we made to facilitate the learning process for today. So we have a bioactivity model. One of them we already explored yesterday, this EOS 3804, which particularly predicts activity against Acinetobacter baumannii, if you recall. Then there is another model that it has the identifier 4E40, which predicts broad spectrum antibiotic activity. We have selected the cytotoxicity model for human cells. We have selected models that are related to ADME properties. We have selected models related to synthetic accessibility and models related to chemical space. What we are going to be doing now is go through each one of the publications of this model. We have on purpose selected models that are well published, that are peer reviewed, so that we can actually have a good learning experience from them. It doesn't mean you cannot use other models. It doesn't mean these are the best, okay? This is the exercise we have selected for you all. So we're going to walk through together through the main characteristics of each one of these models, identifying what are they useful for, what are they not useful for, and any limitations we may find from this. So let's go to it. All the publications are available in our resources platform page, but I would advise you to just for the next 20 minutes, stay with me and let's go through together through the main figures of these publications. And at the end or during the session or after, you can really go and do a deep dive on these publications to learn more about these models, if that is what interests you, okay? So bioactivity, let's start with the most important feature that we want to obtain from our molecules, because we want molecules that are active. We have a very good model that predicts A baumannii activity, but because pathogens, some of them tend to be similar, especially in this AMR space, it doesn't hurt to assess your molecules against more than one endpoint or more than one strain, to basically see if our molecule could potentially be an inhibitor of not a very particular strain, but maybe more broadly of other pathogens or drug-resistant strains and so on. So in this case, in addition to the original A baumannii model that we already ran yesterday, we thought maybe it's interesting to check broad spectrum antibiotic activity. Okay, so we selected this paper from 2020, where basically they propose a deep learning approach to antibiotic discovery. Right, great, the title sounds exciting. Again, I've taken the main figure of the paper to just highlight an example. So figure 2 gives all the information in principle we need to understand this model and to be able to apply it to our own research. As we did yesterday, let's stop here 1 second and look at what each of these panels is telling us. 4, the first row, figures A and B, okay? What we have here is, okay, primary screening data for growth inhibition of E. coli, okay? So now we know that what we are measuring is growth inhibition of E. coli. Okay, so that is the target organism. It means that if our molecules are not active against E. coli, doesn't mean they are not active against A. baumannii. Okay, this is an additional checkpoint. So we are measuring E. coli inhibition. We may want to look into the paper which specific strain of E. coli. And then they took an internal data set. They did the experiment themselves. 2,500 molecules were tested with some natural products as well. So this is telling me that this model may actually have some natural product information, so it may do perform well in natural product space, right? Because training data was also from the natural product space. So here they show us the relative growth. In blue, the non-growth, the non-active because there is growth of E. coli. In red, actives because there is literally no growth of E. coli. Fantastic. Then plot B, this is very obvious. We went over this yesterday, but again, this is an area under the ROC curve telling us the model performance. And we can see how the area under the curve is very close to 0.9. That's a very good model. We can see how it goes fastly up before going to the right. So again, the shape of this curve is something we like. So we have a model that has been trained on internal data, is based on E. coli growth inhibition, and it has a good performance. Fantastic. If now we go and look at the rest of the plots, well, C, D, and E, what we have here is rank order prediction scores of the drug repurposing hub molecules that were not present in the training data set, top predictions from the data in here, so from the drug repurposing hubs, so something that they are not telling us here, but if you read through the paper, you'll find it very quickly, is they build a model, then they use the drug repurposing hub to basically obtain predictions, and then they select from these predictions a few molecules to test. Okay, so they test them and they see how some of them are active and others are inactive. As expected, and not all of the predicted molecules as active are active. Some of the predicted molecules are actually inactive. It's interesting to see here that they do not only select the most active, okay, the top 91 predictions, but they also select the bottom 63 predictions. So what they are doing here is also testing that what is predicted to be negative is an actual negative. Okay, so this is a nice exercise, a good validation of the model. So we know we have this classification model. They select molecules. If you didn't read through, they clearly say cutoff of 0.2. Anything above 0.2 is good in optical density, right? That's the experimental cutoff here. And then they just rank the molecules and select the top 199 and the bottom 63. They extract two measures from here: true positive rate, meaning of all the things that I've predicted positive, how many of them are actual positive? Well, I'm gonna give you guys 1 minute. You just need to type in the chat if 51.5% of a true positive rate is good or bad in this setting. So you just need to type good, bad, and we see what you guys think. Just some answers in 1 minute and then we'll move on. I'm gonna give you, okay, we have a bad. All right. Okay, we have a good here. We have a bad. All right. Good, bad. Okay. So now let's think through this. This true positive rate is not the performance of the model in the train test split, but it's a model validation. So what this true positive rate here is telling me is of all the molecules that you test, 50% of them are going to be actives out of the box before I do anything else, before I look at these molecules at all. So if any of you does drug discovery for this particular problem, which is basically E. coli or hard to target pathogens. Can you tell me how many molecules in a regular screen out of the blue, if you just take a collection of molecules and take 100 random, how many of them will be active? Is that anywhere close to 50? So the question is that 50% a rate that you expect, yes or no? When you just, you take, exactly. So I have the first answer, not at all, just 1 or 2, okay? So this experimental validation with a 50% of true positive rate, it's very good. It's very, very good because it's telling you that 50% of the things you test are gonna be active. This is way more than actually what you find in a real life setting without the help of AI, okay? So of course the models are not perfect, they are not expected to be, they are really just helping you curate good molecules from large data sets, okay? So probably you would only have hidden 1 or 2 of these hits if you randomly select 100 molecules from the drug repurposing hub, and now we hit 50 out of 100, so actually very good. And on the other end of the spectrum, if we look at the negatives, we are only 2 molecules that were positive were actually very downranked by the model, and the rest of them were really negative. So it's not a bad model. It's actually really helping us in the process, right? So this is the type of information you need to be critical about, and you need to think. You don't just stick with the number, 50% doesn't sound very high, because depending on the scenario that we are, 50% is actually very high, okay? So just to give you this critical perspective on how to evaluate whether a model seems to be nice, working, or helping or not. Okay, so this is the first model that we are going to use, which is bioactivity. Okay, we have bioactivity can save money. As we saw yesterday, we have this broad spectrum antibiotic activity. Let's go to the next step, cytotoxicity. This, I think we can all agree. We want to make sure that our molecules kill the pathogen, but don't kill human cells. This we do with this model, which, if you read through the paper, it's also based on antibiotic drug discovery, and they build 2 models. So we are just going to focus on the one that is about cytotoxicity, which is this panel here, okay? So this is figure 1, a lot of information, but basically, what are the key points for this publication is here in the schematic representation, it's telling me that what they did is take almost 40,000 chemically diverse compounds, screen them experimentally, and use this data to train a model, which then was used on a virtual screening cascade of 12 million compounds. Okay, so that is the idea of the paper. We screen models, and we screen molecules, and then we build models. So part 2. Then, for this section here, which talks about cytotoxicity, let's see, they assess cytotoxicity against HepG2, HSKM, and IMR90. These are typical cells to assess cytotoxicity against. So when you use this model, you will get 3 columns as a result, not 1, but 3. The prediction of toxicity in HepG2, in muscle cells, and in lung fibroblasts, okay, in these 3 cells types. Okay, they say this data comes from single point inhibition viability data at 10 micromolar concentration. Yes, instead of giving us the area under the curve, what they give is the area under the precision recall curve. We did not go over this yesterday. The idea is very, very similar. We want numbers that are closer to 1, okay? I just want you to remember this. The area under the precision recall curve has as a baseline not 0.5. Remember, in the area under the raw curve, 0.5 would be random, and the closer we get to 1, the better the model is. In area under the precision recall curve, the baseline is the proportion of positives. So if my data set has only 5% of positives, 0.05 will be my baseline of area under the precision recall curve. So the numbers tend to be much smaller, much farther away from the one, but the bigger they are, the better the model is, okay? So an area under the precision recall curve of 0.33 is not bad. It's not as an area under the curve of 0.33, okay? It's still good. It's a good model. You can read more on areas under the precision recall curve elsewhere, but the idea is just for you to remember it's a measure of model performance. The higher, again, the better. So what we know about this experimental data for cytotoxicity is that they use a 90% viability cutoff, which is very stringent, okay, to really catch things that are cytotoxic. And these are the percentages of data that you got, okay? So 8.5%, 3.8, and 8.8%. So again, this is an imbalanced data set, okay? So they use this data and they train a model and they validate it, and it's a good model, so we can use this to predict cytotoxicity. Again, this is a classifier, so we have already a Baumani classifier, E. coli inhibition classifier, cytotoxicity classifier. And then they use this model. As I said here, I just put this for example here. They use this model on a prospective screening of 12 million compounds. If we look through this graph, basically, and through here, what they say is compounds with less than 0.2, because we want low probability of one, we want molecules that are not active, not toxic. Compounds with low cytotoxicity prediction are retained. So they are really strict in not wanting cytotoxicity, okay? So they use a probability cutoff of 0.2. Okay, so now we already have bioactivity and cytotoxicity. We have all classifier models, pretty straightforward to interpret. Yeah, even though these graphs sound very complex and the papers now may seem complicated to understand, as you can see, they are really not that difficult. You just need to look for the key information in each model. Okay, any relevant questions, or can I proceed, guys? Okay, I get an okay, so I guess I'm proceeding. So remember, on our schema, we have assessed bioactivity, we have assessed cytotoxicity. We may want to know more information. For example, cardiotoxicity, which is one of the reasons why drugs fail in advanced stages. So it's good if we can predict cardiotoxicity. So there is this paper that builds several models, and basically, I just go to the point here. They explain us the data sets they use. They predict plasma protein binding, permeability, hERG inhibition, that's the cardiotoxicity, and ether A-go-go channel, and cytochrome inhibition. I took this information from the publication. We are just focusing on the hERG inhibition assay. So basically what it tells us, we used data compiled by someone else, okay? So you may want to go to the original publication and read more about this, but basically what they do is provide almost 7,000 compounds for which we have IC50 values, okay? The measure of IC50 in this case is in nanomolar, and what they tell us is we transform this into PIC50, not IC50, but PIC50. They train, as it can be seen here, a regression. So what the regression is going to predict is the label that we fed into it. It is a PIC50, not IC50, and I'm going to, again, ask someone to please drop in the chat what is the PIC50. Not IC50, but PIC50. I know someone knows this. Okay, minus logarithm of the IC50. Okay, so the values are reversed. Yes. So let's be conscious about that. That is why it's so important to understand the data that has been used, because if I get the values and I assume they are IC50s, I will get the entire model wrong. This model is going to predict PIC50 values. Okay. And the last thing that they tell me is, okay, the R values range between 0.53 and 0.74 for the 3 regression models. They are not perfect, but they are better than random. Okay, so more or less the models are validated, and it's a good model that we can apply. Okay, remember it's a regression, so we won't get a 0 to 1 number, but we will get an exact predicted PIC50. Great. Let's move on because I want to have time to discuss the exercise. If you recall, we are on the third slot of our circle. So now we are going to admit properties. Cardiotoxicity is one of the admit properties on the toxicity end, but there are many more things that we may want to assess. I just want to put it out there. I'm sure many of you have already used platforms that calculate AI. Something that we really like is this new Admit AI platform. We have incorporated these models in the hub. And basically, this is an entire platform that, importantly, it's trained on 41 data sets, 10 regression models, 31 classifier models from this Therapeutic Data Commons initiative. So if you run a prediction for this model, you will not get one column, you will get 41 columns, one for each admit data set. They tell us a bit of information about the model. They did a train validation test split 5-fold, so 5-fold cross validation, and they use an ensemble of models. Great, fantastic. We just see that they did build the models, and they have a good performance, R squared over 0.6 for 5 of the 10 regressions, and area under the curve over 0.85 for 20 of the 31 classification datasets. You can go through the paper to read more in detail about each specific endpoint. But the idea is that this ADMET AI is built on a benchmark. Therapeutic Data Commons is a benchmark, is a data that has been specifically curated for machine learning. It combines classification and regression tasks, and it gives a complex outcome because it has 41 columns. Okay, so if you run this model, don't be overwhelmed. There is a lot of endpoints that you may want to consider, and some of them may be relevant for you, some others may be not so relevant. Okay, so this is where some critical thinking is needed to see if we want to take into account the 41 columns or we just want one of the outputs of this. So let's go on to the end of our round. So we said we, as chemists, are maybe interested into whether these molecules can be accessed in the laboratory, can be synthesized in the laboratory. There is several models that do help you predict that. We just chose one actually quite old from 2009, just because it's nice and it's easy to use. So if just by looking at this graph, you can already see what this model is doing. It's giving us a synthetic accessibility score. We have 3 data sets here: natural products, bioactive molecules, catalog molecules. Without even reading the paper, if I use this model, it's very easy. Natural products have a high score. Natural products are difficult to synthesize. Bioactive molecules have a lower score, and catalog molecules, by catalog, we mean these molecules that you can pre-purchase that are synthetic libraries, have very low scores. So a low score in this model means that it's easy to synthesize. A high score means that it's difficult. And if you want to read through the model, they use a very simple approach in which they calculate the synthetic accessibility score for their training data set, which is a combination of a fragment score and a complexity score, right? So a fragment score is basically how common the fragments that these molecules have are found in, for example, PubChem. If the fragments are very common, it means that probably this molecule is easy to synthesize, right? So that's one thing that you take into account. The other thing that you take into account is the number of complex features, this complexity score, which is, for example, if you are very big, that penalizes you. If you have a lot of microcycles, that penalizes you, etc., etc., right? So they build this training data to calculate the synthetic accessibility score and then basically train a model to predict it for new molecules. How do they validate it? That's interesting. That's why I put here the second graph. They say here chemist score. So they ask chemists, humans, to take 40 molecules and basically predict what the score they would give to these molecules. In blue is the score that the chemists gave to the molecules, and in red is the score that the model gave to the molecules. So as you can see, they are pretty much aligned. This manual validation done by a chemist eye is pretty good of this model. So basically in this model, we have a value that is going to tell me the synthetic accessibility. The lower the value, the more synthetic accessibility, okay? There is a lot of other models that do this. This is just part of them, one of them, an example, okay? And finally, and just on time, let's go to chemical space exploration. As we saw the other day, we can use PCAs and UMAPs. We can also do something much more simple. For example, what we chose to present here is a natural product score to assess how similar or how natural product-like our molecules are, if they fall in this chemical space or not. So again, this is very easy to interpret. We have this natural product likeness score that the model predicts. We see coconut having high scores, and then we see zinc having low scores. Coconut are natural products, zinc are catalog products, so obviously high scores indicate more natural product likeness, and they compare it with the score that we've just seen, the synthetic accessibility score in this publication, which of course natural product likeness correlates with synthetic accessibility. Natural product-like compounds are more difficult to basically synthesize than synthetic compounds. So no need to understand much more. There is a lot of information. You can go there, but basically they train this model based on coconut and zinc. And what they try to learn is whether molecules are more natural product-like or more synthetic product-like, right? So this is pertaining to which chemical space our molecules belong. Okay, I'm gonna take a pause here because this was a lot. So do we have questions that we need to address before moving on, guys? Mikel or Jason?
Maybe just one quick one. What is the importance of the natural product likeness score and hit to lead prioritization? So how early do you want to bring it in?
Excellent. Thanks, Bryce. So these are just examples of things you could look for. It doesn't mean necessarily that they are all essential, especially you need to think about your particular use case. In this case, natural product likeness score is probably interesting in addition to synthetic accessibility. If you have a large data set in which you suspect you may have natural products, you may want to use this model to confirm it. You may just want to use it as a representation of the chemical space that you're studying, not necessarily for the prioritization of compounds, okay? So it is not that you have to use all these models always. This is just an example. This is just to have some discussion, okay? So you just, can use it or cannot use it. It really depends on the use case that you have. If you have manually curated a data set from natural products, you may use just this model to reinforce, like, look, these are natural products. They score high in this model, etc. Okay, so it's by no means set in stone that you have to use these 4 models that I presented. On the contrary, there is a lot of models out there. This is just an activity. I hope this is clear to everyone. So I don't see the screen, but I hope there is some thumbs up from participants about this. Okay, this is an exercise for the class. We chose a few models that allow us to discuss the concepts we've seen: classification performances, cutoffs, and so on and so forth. Okay, is that clear? I hope there is some thumbs up on the screen. Yeah, okay. So I'm gonna stop sharing for a second because I want to make sure that we then go all into the same page. Let me stop sharing and let me now share this screen. Okay. So I hope by now you're all very, very familiar with this web page, which has all the course contents. Okay. So the important part here is course contents and resources. I will ask you to do, please, really, if you can unfollow my lead here and really do what we ask for the exercise, it will really help for all of us to complete the exercise, okay, without technical issues. On day 2, because that we did yesterday, we have the data set for activity. If you don't have this downloaded, please go ahead and download it. This is just the list of smiles that we are using. This is 100 smiles. It's ready to input into Ercilia. You don't have to do anything. You just download the CSV, put it directly in the platform. Okay, that's the data set we are going to use today as well. If yesterday you are in the group where you had the models crash and you haven't had time to rerun or download the results, you can just go ahead and click in this link. It will give you the results of yesterday, what the model gave you, this prediction of Acinetobacter baumannii. You can download these 2 files and keep them with you in your computer for day 2, okay, day 3, yeah? So please do that and that. And under resources, if you're still feeling lost, just go to the shared folder. You will find the publications named, okay, let me just show it. You will find the publications named by its identifier. I'm gonna leave that on the screen at the end so you can see, but basically you can look through all these publications, okay? What I'm gonna ask you to do now, and stick to it, okay, of everything I have presented, except for EOS 3804, of course, that we saw yesterday, I'm gonna ask you to individually choose one of these models, just one of them, and stick with it throughout the rest of the exercise, okay? If you try to tackle all the models, you will not have time. You will just have 100 jobs running submitted to all the models. So just pick one and do a good exercise with only one of them, okay? Doesn't matter which one, the one you like most, the one you like less, the one you find easier to interpret, the one you find harder to interpret because you want to challenge yourselves. Choose one of them and stick to it for the second part of the exercise, which is going to be this. And now let me share again what we are going to do just now. Okay. Breakout activity again. Log into the Ercilia Model Hub platform. If you saved your generated ID, you please use the same, because there you will find the model runs that you are doing. Okay, so you can log in back with the same ID. If you didn't manage, you can generate a new ID. Make sure you have yesterday's data set, the one that I shared, not anything that you have modified or that you have added new columns to, okay? Just the 100 molecules, that's important. Download the predictions for the previous day if you don't have them already, okay? So that you have what we did in the previous day. And then choose one model, the one you like more or you like less, and run predictions using these 100 molecules for this additional model, okay? And then you should use 10 minutes to think. Even while the model is running, you can already start thinking, how would you use the A baumannii combined with at least another property, okay? Combined with at least another property to select molecules, okay? I'm gonna give you an example, a very clear example. If I have, maybe I should, oh, okay. I'm gonna just stop sharing and go back to this screen where all the models are so that people can see. That will be easier. Just give me 1 second. Okay, this screen, I think that's gonna be easier. I should have copied that. Okay. If I say, okay, so I have EOS 3804, and my chosen model now for the second part of the exercise is for E40, which is E. coli antibiotic activity. I'm gonna make a prediction. I'm gonna put in a single Excel file my molecules, one prediction, the other prediction. And then obviously, if a molecule is active, what is better, a molecule that is active only against A. baumannii or predicted active against A. baumannii and in this broad spectrum antibiotic model. So probably if I have to select one molecule, I will select the one that is active in both models, right? If we are talking about cytotoxicity, if a molecule is active in the cytotoxic model, probably I don't want it. Or if it has a PIC50 value that I don't like, probably I don't want it, okay? Or it's not synthetically accessible or is synthetically accessible. So the idea now is to make these combinations, okay? Let's, each one of you just focus on one other model so that we keep it time constrained and resource constrained. And then we will discuss this back at 1:15, okay? 1:15, we come back together. I'm gonna talk about all these models briefly, so it doesn't matter which one you choose for the activity. Now it's just for you to think individually, so you get some practice time, and then I'm gonna go over all of them. So don't worry, whichever you choose, it's good. All right, I'm gonna leave that on screen so that you know what model corresponds to what, and I'm gonna mute myself and wait until 1:15, okay? 13:15. If the models take time, some of them are very large and may take a lot of time running. Don't worry, you can complete the exercise later. Just stay with us, read through the publication, make sure you understand what we are doing, and you can complete the exercise in the afternoon. Okay, some models, I warn you, take more than 10 minutes to calculate, just because they are very large models. There is not much we can do about this, okay? So don't worry if your results take time. I'm gonna stop here. Okay, I see a couple of people confused with the exercise, so let me go over this again. Okay, hope you're listening to me. You have a data set of 100 molecules. What we are asking you to do is to make a prediction of these 100 molecules for one of the examples that we've gone over. Okay, it can be cytotoxicity, it can be bioactivity, it can be whatever it is. Then, based on these predictions, we are going to discuss which would be the best molecules to test in the laboratory, how to prioritize molecules for experimental testing based on the predictions that we get from these models. We are not going to do this only based on one model. We are going to do this based on the bioactivity predicted yesterday and at least the second model that you can now choose and run. Okay, so this is the activity. If you want to go through the slides again, the slides are already linked, so you can go through the slides. If you have questions, please drop them in the chat. I see some hand raised, but please drop the question in the chat, and we are going to answer it there. If you prefer, I can only show one, so I either show the model identifiers, or I can show the breakout activity slide if you prefer. Sure, I can do that. Okay, this is the activity, but again, you don't have to complete it now. You don't have to worry about the final submission. Just play with it, okay? The platform link again is in the Git book. Please use this resource to find all the links, okay? Under the bottom part, resources. To create an Arcilia account at the moment, just click on Generate ID. There is 3 buttons when you enter the Arcilia platform. On the first one, it will generate a random ID that you can use to log in. It should be straightforward. And just go through the AI2050 online, where you have all the links to the hub, links to the publications, links to the slides, okay? So we are going to give people a few more minutes. You can, yes, Victor. So just do one, and just submit. I will see that by the name of the column, or you can just add an explanation. Just do one of them, because of course we don't have time to do all of them. I'm going to show the results for all of them, okay? So don't worry. Okay, folks, so because I want to have time to go through all the models and answer all the questions that may arise, let's please start making your way here to the main screen, okay? Where we are gonna all together discuss model by model, whether it is informative or not for our use case and whether we want to use it and how. I will ask you again, I will ask very clear questions like yes or no, high or low. And I would love to see you answering in the chat so that we can guess more or less if you're following the content or not. So come back here. You'll have time to play with your Celia platform, especially when it's fully released in December. So come back here on the main screen and let's go over these results together, okay? So I'm gonna stop sharing the presentation and then we are going to share the results. I know some of you were having issues yesterday into how we analyze the results of the hub. So I'm going to go over this again to start with, okay? I use Google and Google Sheets, which is exactly the same as if you use Excel, okay? I use that because I'm on a MacBook that doesn't have Excel. So let's go to your Excel or to your Google Drive. This is what I would do now if I have to analyze the results, okay? I have put all my results, which are CSV files, in this folder. So let's start. I'm gonna create a new file, okay, a new Excel file. I'm gonna name this my results, fantastic. And I know the first column I want to have is my smiles, my molecules, right? These, okay, so I have my results and I have a first column that is gonna be my results, my smiles. This I get from the CSV file that hopefully you all downloaded, which has all these smiles, which I can copy very nicely. So I'm gonna copy that and I'm simply gonna paste them in order, okay? Here it's very important that we keep the order. I'm gonna paste them in the first column so we have our input. Then the first prediction that we made, if you recall, was model EOS 3804. It's again a CSV file. If you open it in an Excel file, it will open directly. So here we have the column name. This is important because it's telling me which activity we were using. Yes, in this case, A1 inhibition. I have my predictions. I'm gonna copy those again. I go back to my Excel and I paste them, yeah? So now I have my smiles and my A1 inhibition probability. I go back to my results and then I go next. If I did the prediction for EOS for E40, I'm gonna open this file and basically, again, copy. In this case, we only have one column, which is inhibition at 50 micromolar of what? Remember, this model is E. coli, so this is what we get. So I'm gonna copy these results again in order, again, not modifying anything, and pasting them here. So we already have smiles, A1 inhibition, and inhibition at 50 micromolar, okay? The other model, the next model that we talked about was cytotoxicity. Cytotoxicity produced this other CSV file. This model takes, it's a very big model, so it takes time to calculate. So if you selected that, you may not have the results yet, just wait for it. So let's just select the 3 columns and again, copy them directly, concatenate them in order into my CSV Excel file, okay? So hopefully it's clear how to do this. So if this is more or less clear and you understand the idea of concatenating the results one after the other so that we can analyze the results of several of the models, not just a single outcome. I hope just say yes in the chat so that we can see that you are following. Even if you didn't manage to finish the exercise, it's fine. We are here to learn, so I hope you could follow me. Okay, yes, great. I see a lot of yeses. Excellent. So this is how you basically concatenate results. Once you have all the results, okay, I've already done that, so I'm not gonna do that in the screen. But basically, then in Excel, you can easily select. Then, if you know Python, by all means, please do that with Python and Pandas and whatever package you use. I'm just really now here being for people who is non-programmers, okay? I can select the first column, the first row, the columns, and create this create filter. With these filters, what I can do is I can sort molecules, and it will rearrange the entire rows, okay? So if we look here, this row will remain with, of course, the molecule and its correct predicted values, even if it gets sorted. So if I sort A to Z, it puts the lowest value first and it has rearranged the columns. If I sort Z to A, it puts highest value first and it has rearranged the columns. Right? So this is now very easy because we can rearrange the columns and see if we want high activities or low activities. I think we can all agree that the most important activity, the most important thing that we want to optimize for in this drug discovery exercise is the activity against our main target. Can someone type in the chat what is, of all these columns, which one is the most important one of all these prediction columns? So, okay, we've got a couple of answers. Column B, bioactivity, inhibition, okay. Okay, so let's look at the columns again. The smiles, obviously, it's important because these are our smiles. But then what we really want to optimize for at the moment is column B, which is A-Baumani inhibition probability. This is the one that we really, really have. So I'm going to keep my molecules ordered based on probability of inhibition of A-Baumani. Then my question for the chat. Do we want this value to be high or low? This column B. High, yes? We want high because we are doing a probability of inhibition, so we want the probability of inhibition as high as possible. So that is why I have sorted this column as high, okay? Just to make it nicer to see, let me move to this other file that I prepared for you guys, where I did not only concatenate the results of these 2 models, but all of them, okay? Already all the models that we've discussed now. And I've gone a step further. Again, this only please if you are knowledgeable of Excel. I've gone a step further and colored some of the columns. This column I've colored so that higher the number, the more green it is. So that's very obvious. We want molecules that are the greener the possible, okay? So I think yesterday we said these 10 first molecules look very good because they have a probability score of over 0.2, which is what the authors defined in the publication from yesterday, okay? So I really like these 10 molecules. Maybe some here that have lower values may be interesting or not. But this is basically my first choice. If I would come to you and say, that's great, but you can only choose one of them, of these 10. Let's see what else would we use to decide. The second thing we've predicted is inhibition at 50 micromolar of E. coli. Again, do we want this value to be as high or low? This second column, inhibition at 50 micromolar. Yes, we want it to be high. Exactly. We want it to be high, as high as possible. Does it mean that if it's predicted to be active against ABO money and predicted to be not predicted to be active against this second model, that the molecule is not good? No, it does not mean that. Okay, because the second column is a broad spectrum antibiotic activity, is not really my endpoint. It's a help model. Okay, so I may want to consider it or not. Okay, for example, I see down here a molecule that has a very good value, but has actually not a really good value of predicted against my particular pathogen. Okay, so this model may not be so interesting. It's true that here this molecule and this molecule has somewhat high values, but before I make a decision to commit to maybe this molecule or this molecule, let's look at this very interesting column here on the third. Okay, these three columns are cytotoxicity. This is what you get if you run model 42EZ. Let's focus only on one of them, okay, just to make the exercise easy. Let's focus only on HepG2 toxicity. Let's forget about these other two, okay? I'm actually gonna delete them just for simplicity, okay, for the exercise we are doing now, just to focus on one of them, on one toxicity endpoint. So cytotoxicity. Can you type in the chat if you want this value to be high or low? Okay, Bryce says low, 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 exactly. We are toxicity, so we want this to be low. So that's why I colored this in red, because I don't like this molecule at all. It has a good predicted bioactivity. It has also inhibition in another bioactivity model, but it's very toxic apparently. So actually, I don't like this one so much. Maybe if I had only, again, this is a made up exercise, but if I only had one shot, maybe I don't want this molecule and this molecule and this molecule, because they may have activity, but if they have basically cytotoxic activity, it's not excellent for me. It's not good. I don't like it. I don't want them. Yeah. So I would probably not select this one as my top candidate, neither this one or this one. Regardless of their bioactivity, very high toxicity makes it clear that they will not progress in the drug discovery screen. Yeah. Okay, let's go to this next column. Synthetic accessibility score. Do we want a high value or a low value? Okay, there is some in between discussions, high or low, synthetic accessibility score. If you go back to the publication, what you will see is that these bioactive molecules and catalog molecules were getting numbers around 2, 3, whereas natural products were getting numbers around 4, 5, 6. So I want low values. I want values around the 2s or 3s. So based on this model, most of my top molecules seem to be good. I have some 4s and 5s down there, but again, this is mostly a synthetic small data set that we have curated. So they are good. They seem to be easily accessible. We can draw them. We can see how they actually are. We can ask a chemist to have a look at them. But overall, they all seem good. This would not, I don't see any molecule that I would discard based on that. Okay. Another column, PIC50. Okay. And now here is where it gets more tricky. This is cardiotoxicity. It's a regression exercise where we don't get the probability, but the actual predicted value. In this one, which is cardiotoxicity in PIC50, do we want values that are high or low? High or low? I'm giving some time for people to think. Okay, let's see what pops up in the chat. Some highs, some lows. Let's clarify this. Okay. It's cardiotoxicity, so we want molecules that are not active, that are not toxic. if we look at the IC50 value, IC50, we want an IC50 that is high, right? That you need to give a lot of concentration to actually have an effect. Maybe above 10 micromolar, maybe better 20 micromolar, right? Because the PIC50 is the minus log, it goes inverse. So if we want a high IC50, we want a low PIC50, a low PIC50. Just to give you some clear numbers, let me type here in the chat. 1 micromolar is a PIC50 value, and 10 micromolar is equal to 5 PIC50s. These you can revise on your own, do the maths, look online, but basically, sorry, for, what? Let me edit this. For PIC50. Yeah, so anything below 4 I like, and you can see how it actually molecules that have a high cytotoxicity also seem to be quite cardiotoxic. Like this molecule here has a very high PIC50. It's predicted to be cardiotoxic, so it's really something I'm not interested in. Okay, I would much rather go to molecules that are lower. Yeah, so we've gone through bioactivity, cytotoxicity, synthetic accessibility score, PIC50. All these columns here relate to what? ADME properties. These were calculated if you run the EOS 7M30. This is ADME AI. It gives a lot of columns. Things that you may be familiar with is the Lipinski rule. So how many of the Lipinski rules these molecules are following, and then inhibition, penetration of the blood-brain barrier, inhibition of the cytochromes. Okay, so this is a lot of extra things that you can look for and decide if you want to use them or not. Okay, this is a lot of things. I cannot comment on all of them. Perhaps very interesting is solubility. So I let you go through. This is very well documented. Again, it's a benchmark data set, so you can look at what each of these columns mean. Okay, but because we are getting to the end of the session, let me just end up stopping here. Natural product scores. I see some of you posting in the chat, I got negative values. Exactly. If you look at this publication, you will see how the natural product score goes from minus, for example, minus 15, minus 20 to plus 20. Molecules that were on the zinc dataset that were non-natural products were below 0. Molecules that were natural products were above 0. So this is just confirming what we already discussed, that this data set that we provided you with is mostly composed of synthetic molecules. So that's why you get mostly negative values, okay? For our particular exercise, I don't think this is much relevant. Maybe some of the properties here are relevant, so it should be interesting to look into them in the same way that we've looked into this. For this particular exercise, Bowman activity seems the most important one, so definitely these molecules. I would maybe discard the ones that have high cytotoxicity or high cardiotoxicity. This E. coli model is not giving me a lot of extra information, so it doesn't seem that my molecules are broad spectrum antibiotics, but mostly predicted to be active only in one pathogen, which is fine. It's most of the cases, and they all seem reasonably well accessible in the laboratory. So I would not discard any molecule based on that, okay? So this is how you can use several models to get information about your molecules that is not restricted to bioactivity, but you can really make a full assessment of the data sets that you have in front of you, okay, very easily. And through Ercilia, using Excel, if you are a more advanced user, through basically the CLI, so programming and so on, and doing Python analysis of the data, okay? So I hope this is useful. You can continue playing with the hub. As we said, this is going to be fully deployed by the end of the year. And there you will be able to run more models than the ones that are just there for the practice courses. And I encourage you all to just have a look at the hub, at the models that are online, and the ones that may potentially be online, read through the publications, and familiarize yourself with all this world of biomodel, bioactivity prediction, and etc. So with that, we just get to the really end of the session. And that's it. So Juliana, on the ATME, so these ATME endpoints, these are really related to drug discovery, basic drug discovery courses. So I encourage you to have a look at the publication that explains all the ATME datasets that were used, and at the HCD Foundation webinars on basic drug discovery properties, where you will learn everything about ATME, okay? The model just makes predictions on different endpoints. What you need to do here is really do drug discovery basic exercises to understand what they mean. Okay.
Gemma, thank you so much for that really fantastically clear presentation. I think let's give Gemma a massive round of applause, and Miguel and Jason for harnessing the chat and answering where possible. Longer answers will be provided via email as usual. Guys, thank you for your fantastic engagement today. It was wonderful to see you engaging with the chat and really trying to figure this stuff out. Tomorrow, Miguel will be taking the lead in looking into the future and thinking about what's next in this field. And we'll also have a slightly longer Q&A session tomorrow. So a lot of the real pertinent questions that have come up this week are going to be addressed in a little bit more detail, and there'll be a little bit of opportunity to ask some questions live as well. Okay, so don't miss tomorrow. We look forward to having you. All the links are in the Gitbook as usual. And also I'll be sharing the recording later today. Thank you so much, everybody. See you tomorrow.
Thanks, Nicola. Thanks, everyone.